Hello, welcome back. So, last section we talked about prokaryotes and eukaryotes. Now we're going to talk about eukaryotic organelles or cellular organelles. I briefly mentioned what organelles are in the last section, but we're going to dive into them and look at all the different organelles. So, before we get to organelles, let's talk about all the different types. Excuse me. Let's talk about what organelles are. Organelles are known as little organs. They're small structures within the cells that perform specific functions. I like to think of them in two ways. One is you can think of them as different rooms within a house. The kitchen is the room where you prepare food and make food. The bathroom is where you go to the restroom and shower. Your dining room is where you eat, and so forth. Each room within a house performs a certain function. So each organelle also performs a very specific function within the cell. Another way to think about this is that they're miniature organs, just like our organs. How we have our lungs to breathe, we have our brains to think and process information, we have our heart to pump blood, and so forth. Each organelle in this list has a very specific function that it does within the cell to help the cell function. So in this section, we're going to go over all the organelles within a cell. The key thing I want you to know, because I'm about to give you a lot of information, is know the function of each one. As long as you know the general function of each organelle, that is all you need to know. So the first one we're going to go over is the cell membrane. This is the one you will need to know the most, including the function, structure, and the proteins in the membrane, very briefly. So the function of the cell membrane, and keep in mind the cell membrane, like we talked about last time, is the structure that surrounds the cell. It's the structure that separates the inside of the cell, the cytoplasm, from the outside. Now, as the kind of the barrier that separates the inside and the outside of the cell, it has two functions: separation and protection from the environment, meaning that it acts as a barrier between the inside of the cell and the outside of the cell. It prevents things going in and out, or certain things going in and out. And that leads to the next thing: transportation within and outside the cell. So the cell membrane is what controls what can go into the cell and what can come out of the cell. It makes sure that nothing harmful is entering the cell if it can prevent it, and nothing is leaving the cell that the cell needs. So the cell membrane is for separation from the environment, from protection from the environment, and transportation within and outside the cell. A lot of this we're going to see later in chapter 4 too. So right now I'm going over very, very briefly, and we'll revisit this in chapter 4. Next is the structure. So the structure of the cell membrane is very unique. It's made of what's called phospholipids, something that we briefly talked about in chapter 2. Phospholipids are very special. They are composed of two parts, what's called a hydrophilic or water-loving head, and a hydrophobic or water-fearing head. The way I remember this is hydro means water, so hydrophilic, hydrophobic, both have to do with water, and phobic is fear. So we have a phobia of something, a fear of spiders, a fear of snakes, a fear of the dark. Remember, that's a phobia. So hydro, water, phobic, fear. So this hydrophobic is a fear of water. So if hydrophobic is a fear of water, hydrophilic is a loving water. Philic means to love, or translates to love. So I bring this up because if we look at what the cell membrane is made of, the phospholipids, you can see these hydrophilic heads, these heads that really like water, and the hydrophobic tails that really hate water. So when the phospholipids come together, they form what's called a phospholipid bilayer. A phospholipid bilayer means it's a two-layer, bi means two, so two layers of phospholipids. And they form that because all these little tails, these hydrophobic tails, want to stay away from the water, while the hydrophilic heads want to be near the water. So these molecules naturally tend to form this phospholipid bilayer. These two layers of molecules, with the inside being hydrophobic and the outside being hydrophilic. So next is the proteins. And I'm going to go over this very briefly because we will go over this again in chapter 2, or sorry, unit 2. In addition to phospholipids, cell membranes have a handful of different proteins. We're going to look at 5 different categories of them: transport proteins, enzymes, recognition proteins, adhesion proteins, and receptor proteins. Transport proteins are proteins that are found that function to help different atoms and molecules move within or outside the cell. They are the molecules that help push things into the cell and help push things out of the cell. And you can kind of see the example in this picture here, where 2 of these molecules will go into the transport protein and eventually are let into the cell. It could be the opposite. They can move from inside to outside as well. So remember, transport proteins are proteins that help move things across the membrane. Enzymes. Enzymes are proteins that aid in chemical reactions. We'll go over this more in depth in unit 2. So they are found within the cell, they are found outside the cell, and they can be part of the membrane. So they simply aid in chemical reactions, the changing of one molecule to another. The recognition proteins are proteins that serve as name tags. These are the proteins on cells that state, hey, I'm this type of cell. And us as humans, we do have these proteins in our cells that state, hey, I'm a part of you, I'm your cell. Please don't hurt me, harm me in any way. And this is important because for our immune system goes around and checks cells to see if they're ours or not. If our cells did not have recognition proteins, then we wouldn't be able to detect what's our cell versus what's an invader, and that can lead to a whole host of issues. We next have adhesion proteins. These are proteins that make up the membrane that allow the cells to stick together to different things, mostly to one another. So there are different types of adhesion proteins that help cells stick together very tightly. You can kind of see an example up here where you have 2 proteins, adhesion proteins on 2 different membranes that will stick together to attach these 2 cells. And lastly is receptor proteins. Receptor proteins are proteins that can receive or bind to molecules in the environment. What they do is they can bind to something in the environment, they can sense something in the environment, and that can cause a message to be sent into the cell. So you can kind of see here where these purple circles are the message. This right here is a receptor protein. When that purple circle binds or hits that receptor protein, it's going to send some type of signal within the cell. Tell the cell, hey, the signal just came in, this message just came in, we need to do something. So these are the 5 different proteins within the cell membrane: transport protein, enzymes, recognition proteins, adhesion proteins, and receptor proteins. Make sure you know their general function. Now, we are going to start going over all the different organelles. There are a lot of organelles we're going to go over. The big thing is I want you to know their general function for each one, and that is it. So I'm going to go over one. I'm not going to go over super in depth. I just want you to know the general structures. I did classify these into different parts and different categories. And in general, make sure you know their general functions. We're going to first look at the endomembrane system, which is a group of organelles that work together to transport substances or other molecules around the cell. This system includes 4, roughly 5 different organelles: nucleus, endoplasmic reticulum, Golgi apparatus, and vesicles. The nucleus is the organelle that contains the cell's DNA, or the instructions, recipes on how to make proteins. It's the site where protein synthesis or production starts. We'll get more on that in a later unit. So just like we talked about in the last page, the nucleus is what houses the DNA, which are the instructions on how to make the proteins in the cell. We can see in this image, this blue circle is the DNA. So all the DNA in the cell is held right there in eukaryotes. Now, keep in mind, this is the DNA is the instructions on how to make a protein. Because in the next, we have endoplasmic reticulum, smooth and rough. These are sac and tubes composed of membrane, the same membrane as a cell membrane. For the rough endoplasmic reticulum, they are covered by ribosomes, and ribosomes are the factories that make the proteins. So since rough endoplasmic reticulum or rough ER is covered by ribosomes, they aid in the production of proteins. So some proteins are made within this endoplasmic reticulum, and then they can be moved around afterwards. The smooth endoplasmic reticulum, though, does not have ribosomes. They do not synthesize proteins, rather, they synthesize lipids. They also house enzymes that help with detoxifying the cell, enzymes that help remove toxins from the cell or the body. So the rough endoplasmic reticulum aids in protein production and has ribosomes, while the smooth endoplasmic reticulum synthesizes or produces lipids and helps with detoxifying the cell, getting rid of toxins. The Golgi apparatus is the next one. The Golgi apparatus is a stack of flat membrane enclosed sacs. And you can see here, this is the nucleus. We have the rough endoplasmic reticulum with all these circles being the ribosomes. And we have the Golgi apparatus, this stack of what looks like pancakes within the cell. So the Golgi apparatus is known as the processing center. Proteins are sent here to finish their synthesis. So some proteins are not fully functional yet after the rough ER. They are sent to the Golgi apparatus to finish being produced. The proteins are then sorted and packaged to be moved throughout the cell or out of the cell completely. So the way I think of this is the Golgi apparatus is like UPS. It takes all the packages that need to be sent out. It may do a little processing, making sure everything's in order, and then it sends them out to the correct location. The next one is vesicles. Vesicles, this is going to be a short one. They're small membrane enclosed spheres. All they do is they transport materials throughout the cell. So you can see with the Golgi apparatus, part of the Golgi can pinch off, forming this little circle called transport vesicles or just vesicles in general. These vesicles, if the Golgi was like the UPS, the actual location that processes the packages, identifies where they need to go, and sends them on their way, transport vesicles are like the actual mailmen that are driving around in their vehicles to deliver the mail to the correct location. So you can see how with these four organelles, nucleus, endoplasmic reticulum, Golgi apparatus, and vesicles, all of them are used in protein synthesis in some sort of way. We're now going to go into the next parts, which is the cell digestive system. And there's three that we're going to look at: lysosome, peroxisome, and vacuoles. Lysosomes are specialized vesicles that contain digestive enzymes, or enzymes that break down substances. The big function of lysosomes is it's used to break down food, capture bacteria. It's used to break down food, capture bacteria, worn out organelles, and debris. So picture the lysosome as our stomachs. The lysosome will get the material, break it down, and then that after being broken down, that material can then be used throughout the cell for different functions. The peroxisome is the next. Peroxisomes contain enzymes that break down toxic substances. So these are specific organelles that help remove toxins from the cell. The last thing is vacuoles for this section. Vacuoles are a stack of flat membrane. So the vacuole is within plants. So the vacuole is this big open area that serves as a storage site for water and different molecules. It helps with detoxification and so forth. Keep in mind though, the vacuole is only in plants. Animal cells do not have this big structure. The vacuole, since it's so big, it actually helps with the structure of plant cells. It helps make the plant cells nice and large and rigid. Now, the next two organelles are for energy. So we talked about how proteins are made and processed, what the digestive system, and now we're going to talk about energy. So the mitochondria, this is the big one that a lot of people hear of. It is known as the powerhouse of the cell. It produces energy in the form of ATP, which is a specialized molecule within the cells that holds usable energy. So it's kind of like the cell's currency. So, the mitochondria produces energy through a process called cellular respiration. We'll talk more on that in Unit 2. So whenever you hear about mitochondria, think ATP, think energy, think powerhouse of the cell. Next is chloroplast. Chloroplast is only found in plants and some protista. It is a site of photosynthesis, which is the production of sugar, glucose, and other food molecules using sunlight. So cells or animals that can produce their own food, a lot of times they have a chloroplast. Since the chloroplast can take energy from the sun, it can take carbon dioxide and start forming sugars, such as glucose, that can be later broken down to produce ATP. So, the mitochondria produces ATP, it's the powerhouse. The chloroplast produces sugars through photosynthesis. The last organelles we're going to talk about are the cytoskeleton, cilia, and flagella. So the cytoskeleton is a network of protein tracts and tubes. They provide physical support and rigidity to the cell. So the way I think about the cytoskeleton is similar to our skeleton, where our skeleton provides physical support. It provides our bodies to be nice and rigid, where we can then move around without the fear of just falling over. Cytoskeleton is very similar. It provides that physical support. It provides that rigidity to the cell. It also aids in cell division. Whenever that cell divides, the cytoskeleton plays a big role. And it aids in the connecting cells to one another. So the cytoskeleton, again, is kind of the skeleton of the cell. It provides the physical support, rigidity, but it also helps with moving things around the cell division, and connecting cells to one another. The last two structures are cilia and flagella. Both are composed of the cytoskeleton, excuse me, but both are external structures. Cilia are small external structures that can either move the cell or push substances outside the cell. You can see cilia here as these green strand-like hair-like structures. And in bacteria, a lot of times the cilia are there to help move that bacteria around. But in humans, we also have cilia as well. Within our airway, we have cilia that helps push mucus out of the respiratory tract. So cilia are these projections that can move. They either push the cell or help the cell move or push something outside the cell. Flagella, just like we were talking about in the last unit, is a long external structure that is used to move or propel the cell. So these are these long structures. This is one of a prokaryote with the long structures that help move the cell around. In humans and in animals, there's only one cell with a flagella, and that is sperm. So sperm is the only cell that can move. It's the only cell that can use something to move around. It's the only cell that has a flagella. Now, that is all the organelles you need to know. Again, make sure you know each of their functions in general for the upcoming exam.